year when Earl was alive, he would select the 2 keynote speakers, and amongst his many trainees and colleagues that he had met over the decades, several decades before, and I can honestly say I have not been communicating with him in the last 2 or 3 years, but I know that he would support the selection of the 2 keynotes this year, who are clearly sort of meet the what Earl would have considered leaders in not only in their science but in their philosophy of training and education and mentoring and engagement and just being nice people. So Alisa Wolberg is our first keynote speaker, and I'm really delighted to introduce her. She's known to the CBR. She's been here actually before. That's when we lost the pictures, and our photographer does indeed remember that. It was traumatic for her particularly, but we'll have more pictures this time around. And but Alisa, my understanding, I think she grew up in the sort of the New York, New Jersey area, but she really quickly moved to warmer climates, as she hates the cold, to UNC Chapel Hill, really where she has established her career from early days right through to her current position, where she's the vice chair of research in the Department of Pathology. She's a professor there. She's a leading authority in the world of thrombosis, as it applies also to hemostasis and bleeding disorders. She's really well known internationally in all the organizations at ISTH and Journal of Thrombosis Hemostasis. She's an associate editor of Blood Advances. She's really out there as a mentor and a teacher and an educator, and we're happy to have her here today. Alisa, you're on.
All right, thanks, Ed. That was wonderful. It's such an honor to speak here, and I can't believe that I'm a keynote. I mean, it's just amazing. It's surreal, and it's just, I don't even know what to say. It's just absolutely wonderful. Although I do know what to say. The first is an apology. This is the title that's in your program. It's the most generic title. It could be a title of anything that we're hearing today. I put it in there because we had a couple of different stories that were bubbling up over the year, and I wasn't sure what was actually going to make it to the forefront. But in fact, one story did make it to the forefront, and I'll get to that at a part of the talk. So this is the real title of today's talk, Fibrinogen Factor 13 and Venous Thrombosis. And so the second thing is to say, I didn't know if we needed disclosures. I have funding from a few different companies, but the lower 2, Novo Nordisk and CSL Behring, are the ones to highlight because we did get funding, mice, and products from them, and I will be talking about some of the work, although they didn't have any influence on the science. But it's interesting, I didn't think about this until Ed was saying this morning, but the product that I'm going to be talking about is Factor 13 and the recombinant products made by Novo Nordisk, and they got that from Zymogenetics. So in some ways, I'm bringing the story back here, and I did not put the 2 together until, so actually it's kind of cool to be able to do that. All right, so I'll just jump in really quick with one slide on the pathology, although it probably needs no introduction. I'm going to focus on venous thrombosis because it's a major public health concern. It affects 1 to 2 per 1,000 individuals each year on the US, in Canada, and in Europe, and that number goes to about 1 in 100 in people over the age of 55, so there's an age component that we don't yet understand. Up to about 30% of people with VTE die within 30 days of diagnosis. That's usually associated with pulmonary embolism. And even in the surviving patients, up to about half develop long-term complications, including post-thrombotic syndrome or chronic thromboembolic pulmonary hypertension, where the veins just don't return to normal and the scarring and the processes that led to the venous thrombosis become long-term and persistent. And as a result of that, all of the major research organizations or clinically advocating organizations in our field have determined that venous thrombosis is a major area of research interest, with the idea of that translating that work into improved clinical care. So what we do know about venous thrombosis is highlighted in this cartoon that I've built over the years. The idea is that this occurs in the veins, quite obviously, and it initiates in the valve pockets where the blood flow becomes non-laminar and allows for activation of resident endothelial cells. Those endothelial cells begin to express cell adhesion molecules, and the cell adhesion molecules recruit leukocytes and platelets to the region. And then the procoagulant activity that's elaborated by the leukocytes, platelets, and maybe also the endothelial cells facilitate the generation of thrombin. Thrombin then enables the conversion of fibrinogen to fibrin, and the formation of a fibrin net within the vasculature, normally helpful in hemostasis, but within the vasculature becomes problematic because it traps resident cells, and because around 40% of your cells, give or take, man or woman, is red blood cells, what it does is effectively trap a lot of red blood cells, and so these clots are very enriched in those red blood cells that are sometimes known as red clots. Following that process by which that clot initially forms, the platelets that are present in the clot initiate platelet-mediated clot contraction events. They pull on the components of that clot and really consolidate that clot into a tight structure that is impermeable to resident enzymes. And so the fibrinolytic processes that are normally designed to clear fibrin don't, and that clot becomes stable and occludes blood flow. To put some context on this, I'm going to talk through very quickly the drugs that we have in the armamentarium for preventing and treating venous thrombosis. Anticoagulants, all of the ones available on the market in some way reduce thrombin generation activity or reduce thrombin generation and or its activity. And so by reducing thrombin, you reduce the conversion of fibrinogen to fibrin, and then the clot doesn't form. And then clots that have formed, you can give thrombolytics. These facilitate endogenous fibrinolysis, so it breaks up the fibrin, and if the fibrin's broken up, it also doesn't function as a net and therefore can't retain the components of the clot, and you can break up the clot. And of course, because both of these processes, fibrin formation and then the fibrin stability, is required for hemostasis, all of the current available drugs to treat or prevent venous thrombosis are associated with some degree of bleeding risk. And so this is something that we're all currently trying to optimize or improve. All right, so this is the thing that I've been talking about. This is a pulmonary embolism that was retrieved from an individual at UNC autopsy service. You can see this is rather large. It was basically a cast of this person's leg that broke free, migrated, traveled through their heart, and landed in their lugs in a saddle position and killed them instantly. On the left-hand side of this, you see some lighter pink regions. Those regions are rich in platelets, and so we know that there's a strong platelet component, at least in the initiation events leading to venous thrombosis. Wolfgang Bergmeier and I recently co-mentored a graduate student who looked at some of the mechanisms by which platelets mediate the initiation of thrombosis. I'm not going to talk about any of that work today. I'm going to focus on the right-hand side of this thrombus where you see that dark red color. The dark red color is indicative of all the red blood cells that are present that I mentioned. These are red clots. And you can really see that if you cut a cross-section through that tail, you end up with a transmission electron micrograph that looks like this. So the weird gray shapes are actually red blood cells, and they've lost their characteristic discoid appearance and assumed this compressed, weird morphology, and that is indicative of those contractile forces that consolidate that clot and basically squish these things into what we now term polyhedrocytes. In and among the red blood cells, you see speckled material. That is the fibrin. And it looks strange because of the cross-section, but it's like slicing through a bowl of spaghetti. And so some of the fibrin strands are actually seen in cross-section, so they look round, and then others are streaks, and you can see that. So when we look at these two components arranged in this brick and mortar fashion, we speculated that the proximity of these and the fact that they're found together implies some kind of mechanistic relationship. And that's what captivated our attention a number of years ago, stemming at the time mostly from my interest in fibrinogen and fibrin biology. And that's something that we've currently, that we've been pursuing ever since. And this is work that's taken place over the last sort of 15, 20 years or so. All right, so that was my very brief background. And from that background, I'm going to launch into a series of chapters or vignettes where I'm going to describe the questions that we have asked or posed based on that original micrograph there, and how we've addressed those questions, the experiments that we've done, the answers that we've gotten, and how they've motivated subsequent questions. Some of these chapters are going to go kind of quickly because they're established and we published them, and then I'm going to get to a chapter that is our newest work and is currently in press, and so I'll spend a little bit more time on that chapter, and you'll see as we go. All right, so the first vignette that I'm going to cover is the initial discovery that really motivated the chapters I'm presenting today, and that is that factor 13 mediates red blood cell presence in venous thrombi. So I'm going to start with another slide, one last slide of background, and that's because even among sophisticated coagulation audiences, factor 13 is considered sort of the least known clotting factor. And when we realized that our biology had to do with factor 13, we had to go back to the literature and do a lot more learning. So this slide is designed to tell you what we learned when we did that. right, so first, factor 13 is a member of the transglutaminase family. If you look on the internet, this is known as meat glue. I actually really like that terminology. It glues together, connects proteins. And it is used not just by the body, but it has been commandeered for use in culinary gastronomy, and it is in fact material that is applied to strange cuts of meat to hold them together. So if you've had a chicken nugget, you've eaten tissue transglutaminase. If I remove that and get back to the science, factor 13 as one member of this transglutaminase family is found in two compartments. It's found in a cellular compartment that is a homodimer. It has two A subunits, and the A subunits contain the active sites. And the homodimer is made in cells derived from hematopoietic cells. So it's found in macrophages and megakaryocytes, and it's secreted by tissue-resident macrophages. The plasma component, which is actually what I'm going to focus the most on today, is actually a heterotetramer. It has two of those A subunits with the active site, and it has two B subunits. And the B subunits mediate its binding to fibrinogen, and which is how we came across it, right? And the fibrinogen binding site of factor 13 is mediated specifically by gamma chain residues 390 to 396. And we identified those a few years ago, and it's the same in humans and mice, which allows for a lot of translation back and forth. It was actually really fortunate when we made that discovery. All right, factor 13 is activated by thrombin, and that makes that a very convenient partnership with fibrinogen because fibrinogen is converted to fibrin by thrombin. So the same enzyme can do all of these things together at the same place at the same time. Once factor 13 is activated, it cross-links fibrin, and it particularly cross-links the gamma and alpha chains of fibrin. There's gamma, alpha, and beta chains. It cross-links the gamma and the alpha chains. It can also cross-link other proteins to fibrin, including alpha-2 antiplasmin, TAFI, and fibronectin. The net impact of all of that cross-linking, all of that meat gluing, is that it increases the resistance of these fibrin strands to fibrinolysis, so they become resistant to dissolution, and it mechanically strengthens the fibrin, which is actually the easiest thing for me to wrap my head around. It makes it a stronger fiber. So the finding that motivated all of this learning about factor 13 was this one here, and this was made by a former graduate trainee in the lab many years ago, Maria Alleman. She was doing venous thrombosis models, and I'm going to take a little bit of literary license now and skip straight to the actual finding. There was a lot of backstory before we got here. But ultimately, what happened, what she learned, is that if she did a venous thrombosis model in mice, we do this by opening up the mice, ligating below the renal branches of the inferior vena cava, and we sew the mice back up. We open the mice up again about 24 or 48 hours later, and the thrombus is present in the IVC, and you can see that fully distended there in the inferior vena cava. If we do this in wild-type mice, this is what we find. If we do it in factor 13 deficient mice, we see almost no thrombus whatsoever. And the thrombus is smaller, and it's smaller because there's fewer red blood cells, which is the part that caught our attention. That was unusual, and it's not typically found. Now, this finding alone could have reflected the fact that these clots lysed, because this lack of factor 13 would enable these clots to become more susceptible to fibrinolysis. But we did a number of experiments to show that that wasn't the case. This is one of them. We took whole blood from those mice, put it in a tube, and then added tissue factor and recalcified it and allowed the clot to form. And when that happens, you get a small contracted clot sitting in a pool of serum. So you've done this either experimentally or just by cutting yourself at some point over time. And when you do this assay, there's not a fibrinolytic component to this. You have to add a fibrinolysis trigger for that to happen. So when we did this, this is what the clot looks like in the real world. You see the clot, you see a little bit of red blood cells sitting in the bottom of the tube. When we do this in the factor 13 deficient whole blood, you see the clot is much smaller, just like it was in the venous thrombus, and it is smaller because all the red blood cells have basically come out of that clot and then have fallen by gravity and are sitting in a pile in the bottom of the tube. And it's striking, and it's been repeated in many labs ever since. At the time when we had this finding, we wondered, because it was so unusual, that maybe we had happened upon something very strange that was going on in mice, which makes it interesting but not necessarily translational. But through scouring of the literature and talking with colleagues, we learned that that's probably not the case, that this does take place in human blood as well. And this is the experiment that ultimately really convinced us and convinced many other people as well. So this was an experiment we did in collaboration with Wilbur Lamb's group at Emory. And what we did is we labeled the red blood cells in red, and we labeled the fibrin in green. And then we captured the clot contraction as it took place in real time. So the video that you're watching right now is what the edge of the clot is looking like as it's contracting. And the red blood cells you can see are staying with the clot, and they're moving kind of in and out of the plane, so it's hard to track because of where we're focusing. But nonetheless, they're all staying with the clot. If we do the same thing with the presence of an inhibitor of the transglutaminases, including factor 13A, T101, what we saw was very, very different. So the clot contraction still takes place, but the red blood cells don't stay in the clot. And they not only don't stay in the clot, you can see they're actively extruded from the clot. They're squeezed out of the clot, as if the platelet contractile forces are squeezing and the red blood cells are being forcibly extruded. And so this video was not only convincing to us about the mechanism, but actually really drives a lot of the hypotheses that we've been pursuing ever since as we watch this take place and try to understand what it is that's going on. So that takes us straight into the second question, which is, how does factor 13 promote red cell retention in the thrombi? How is it doing it? So this is work done by another former trainee in the lab, Jamie Burns. Jamie speculated at first that factor 13 cross-links the red cells to the clot, and this seemed really logical to us. It took about a year and a half worth of very negative experiments to show that that actually doesn't happen. So factor 13 does not cross-link red cells to the fibrin, and I'm still amazed by this, but the red cell surface is really designed not to interact with things. We know that, and it's really good at resisting this particular cross-link as well. We also learned that red cell retention doesn't require any of those canonical non-fibrin substrates that I mentioned. So in collaboration with Hayu Ni and Joost Myers, we showed that alpha-2 antiplasmin, TAFI, and fibronectin also don't seem to matter. So we eventually discarded this hypothesis in favor of a hypothesis that maybe we should have considered from the start. And that is that maybe what's happening is that factor 13 is cross-linking fibrin, which we know it does. And when it cross-links fibrin, it makes the fibrin be a better net. And when it is a better net, it's better able to retain the things in the clot. So the experiments that we did to test this hypothesis led to a couple of observations, which are pretty interesting. The first is that the denser the network, the more red cells are retained. So it really does work like a net, and we were able to show that. But secondarily, we were able to show that what factor 13A is doing is not increasing network density. The networks look the same. Factor 13 is doing something to the individual fibers themselves that is making this more able to retain red blood cells to the clot. And I'm not going to show the data that we generated in order to test this hypothesis, but I'm going to go straight to the conclusion for time, because I'm scared of Ed. We learned that fibrin alpha chain cross-linking promotes red cell retention in the clots. That is specifically, even though I told you it cross-links gamma chains and alpha chains, it is the alpha chain cross-linking that is important for this phenomenon. This is a video that I like to show. I usually show it earlier in the talk, but it represents the kind of fun things you can do with fibrin. You can take an individual strand of fibrin and drape it across a channel, and you can use an atomic force microscope to pluck it like a guitar string. And when you do that, you can learn all kinds of biophysical measurements of fibrin, like how far can you stretch it without it breaking, what force does it take to stretch it, how far can you stretch it and have it return to its native space without being elongated. And this is experiments that were done by colleagues in the UNC Department of Physics. But the data that I'm going to talk about was generated in part by Martin Guthold's lab at Wake Forest. And we know that factor 13A cross-links those gamma and alpha chains. But what Martin showed is that the alpha chain cross-linking specifically increases the stiffness of these fibrin fibers. It takes more force to distend this and move this out of the way. And so we're still working in collaboration with some of these biophysicists to understand this, but our working concept right now is that the stiffness of those fibrin fibers and the way factor 13 improves the stiffness, enhances the stiffness of the fibers, is what promotes red blood cell retention in the clot. That they're simply too stiff to get out of the way, and that's why this works. So this story so far tells you that the fibrin is really important, but is that it? And I have one slide here to say that is absolutely not it. So this is an image of a scanning electron micrograph that Jamie happened to capture one day when he was cruising around a clot. And what you see here actually is really cool. It reminds me of when an octopus comes out of a Coke bottle. You've seen the YouTube videos of this. They're incredibly deformable, and they have to be deformable to move through the pulmonary vasculature hundreds of thousands of times a day to reoxygenate. So within that, could changes in their red blood cell deformability also influence this process? In collaboration with Rafal Polinsky's lab, we had the opportunity to look at this in the setting of sickle cell disease, where these red cells have definitely lost their deformable characteristics, and they become very stiff and very sickled. And I'm going to just show visually what happens. In those clots, this is from sickle mice, we see that not only do all the red blood cells get retained within the clot, they're really retained in there. But inhibiting factor 13 doesn't change that. So we went from this idea that we might have a treatment for sickle cell disease to realizing that was completely wrong. There's something about the red cell deformability that also influences this property, and that's something that Rafal is continuing to look at, and we look forward to data from him on that. All right, that takes me to the third vignette or third chapter of this talk. I mentioned that there are two compartments. There's a plasma compartment of factor 13 and there's a platelet compartment of factor 13. And we were interested in knowing which of these compartments contribute to the ability to keep these red cells in the clot. So this is work that was done by another former trainee in the lab. This is Saravya Katula. And Saravya did a number of experiments, but the one that I think best illustrates this that I can talk through kind of quickly is this one. So she took factor 13 sufficient and deficient mice, and then she took blood from the mice, fractionated the blood into plasma, platelets, and red cells, and then she could reconstitute a fake whole blood. And in doing so, by mixing and matching these components, she could have the factor 13 present in the plasma, in the platelets, in neither or in both. And then she could trigger clot formation, and we could ask what happens to the clot. So it was a very logical kind of procedure. These are the data that suggest we can do this kind of an experiment. If we have factor 13 present, we get clots that are about 35 milligrams. And if we take away the factor 13, they're smaller, about 25 milligrams in this experiment. If we have factor 13 present in the platelets and only the platelets, we don't rescue this phenotype. But if we have it present in the plasma and only the plasma, we completely rescue this phenotype. And we weren't expecting anything this clean, but it was really shocking. And it was consistent with other data that we had as well, suggesting that it's plasma factor 13 specifically that determines the red cell retention in the clots, and therefore how big these clots are. I have one slide of speculation to ask why in the world that would be. Why would this be so specific? So the idea is both of these compartments have the active site. So they're both completely capable of cross-linking things. But if you look at the bold line there, the plasma material, as I mentioned, circulates bound to fibrinogen. It is fibrinogen's best friend. And it's sitting right there on fibrinogen when all the action's happening. The platelet material is actually located in a really strange compartment. It's not in the alpha granules, which is where platelets usually like to keep their coagulation factors. It's present in the cytoplasm, and we don't know why that is. TFPI is also present in the cytoplasm. We don't know why that is either. So the idea here is that what we think is going on is this drawing again, where when we bring fibrinogen together in a half-staggered conformation to enable that fiber to form, it actually brings factor 13 to exactly the right place at the right time where it can begin the cross-linking activity, not just the gamma chains, but also the alpha chains, and that's what it is that promotes the red cell retention in the clot. And we're still trying to understand that, but we have some data that I cut from this talk for time, but I'm happy to talk about that, to suggest, in fact, that's what it is. It's not just the presence of 13, but the fact that it's bound to fibrinogen that enables this to happen. And it's this really interesting idea how evolution brought these proteins together and really enabled this to take place. It's a very finely tuned system. All right, so that gets us to the fourth chapter, and this is the one I'm going to spend a little bit more time on because this is our newer data. And that is, if the plasma compartment is so critical, then it becomes interesting for us to understand what it is that determines the plasma compartment factor 13 levels and how biology is determining these levels. This ends up being a story that got kind of wild. The idea here in one final background slide is that the plasma compartment is this heterotetramer. 2 A subunits that have the active site, 2 B subunits that enable the active sites to sit down and bind it to fibrinogen, which is really important. The thing here is that the B subunits are made by the hepatocytes in the liver. The A subunits are made by hematopoietic cells and are thought to be resident tissue macrophages that reside in the aorta, and I don't understand that. It's just what's been reported, and it's out there. But what it means is that these 2 subunits are found not only, or are made not only in different cells, they're actually made in different tissues. And they both have to get secreted, and they have to find each other in the plasma, come together, and bind to fibrinogen for all of that magic to take place. And that seems crazy. We know that this has to happen, though, because the half-life of the A subunit is very short. It's like 4 hours. But the half-life of the heterotetramer is sort of 3 days in monkeys, but 9 to 10 in humans. So as a drug, this can be given once a month. It's actually one of the more convenient treatment modalities for bleeding disorders. And so from that, the dogma has been that the B subunits must be very stable, and they stabilize the A subunits, kind of like von Willebrand factor and factor VIII and how these things come together. But there's some interesting data to suggest it is far more complicated than that. So these are older data these are older data that bring all of these things together. So the factor 13A subunits, because they have that short half-life, means that 99% of that material circulates with B. And that's some really nice data that Laszlo Musbeck and Eva Katona generated about 10 years ago. And they have an incredible binding affinity, about 10 to the minus 10th. But there's a twofold molar excess of B subunit in plasma over A. And that doesn't seem that unusual. If it's really stable, it should be there. So the table I'm going to walk through, I think, is kind of confusing. So I've cartooned this on the right for you to see. But you see I've drawn, basically for every one A subunit, there are two B subunits. One is bound to A and one is free. If you look at plasma factor 13 levels from somebody who is congenitally deficient in factor A subunits, the A is missing, logically, but so is half of the B. And if you look at somebody with heterozygous deficiency, one bad copy of A, one normal copy of A, they fall somewhere in the middle. So there's something not only about the B stabilizing the A, but something about the A is determining the B levels. All right, there's more data that really drive this home. These are data from the Mentor 2 trial. This is one of the ones that was, one of the trials that was run to get recombinant factor 13 on the market that Novo did, the product that originally came from Zymogenetics. And what they did is to take A deficient individuals and just give them recombinant A. And then they monitored what happened next. And they published one piece of data, but they held on to a bunch of other data. And we had the opportunity to go back into the clinical trial and look at this. And this is what we saw. And it's been seen by others as well. If you give A, then immediately the A levels go up, and then they half-life away. Very logical. What you expect in a drug trial. When you give the A, it forms complexes with the endogenous B, and so the A2B2 goes up and then half-lifes away. Exactly, that's the mechanism of action of this drug. If you begin to look at what's going on with the free B, this is the material that's not bound to A. So it starts high because they've got some, and then it drops because all the B forms a complex with A. But after a couple days, the free B comes back. It replenishes itself. And what that means is that if you look at total B, it doubles. When you give A, the B levels come up. And that supports the idea that there's some relationship between A as it determines B. It turns out it doesn't just happen in humans, it also happens in mice. So these are data from Aki Ichinose looking at factor 13 deficient mice that may have been made here, or maybe Seattle, I'm not really sure, but very locally enough. And the idea is as they genetically reduced A gene, they got less A, but they also got less B in these mice. And then Jamie Burns, on a more recent project than the original one, confirmed this. So as we genetically reduce A goes away, but so does B. And what this meant is if the mice look like the humans in the congenital changes, maybe we could reproduce the clinical trial in the mice. And that's what Jamie did. So we took the factor 13 A deficient mice and we gave them A. This is just the human product. And then we took time points out and looked in their plasma for how much A was present and how much B was present. And when we do densitometry on these, what we see is really remarkable. As you expect, when you give A to an A deficient mouse, the levels are up and then they half-life away, just like they do in the humans. But look what happens to the B. We give A and the B levels come up. And they actually even more than double because the B levels started so low, but they really come up. So the mice clinical trial looks just like the human clinical trial. The mouse model enabled us to then go in and begin doing some experiments to test the mechanisms. So these are the standard questions you ask when you see this kind of phenomenon going on. First, is A needed to release intracellular stores of B? Does the A come, tickle the hepatocyte, and the B pops out? Does A induce B production? Does A tickle the hepatocyte and induce transcription or translation of existing message that would result in this dramatic increase in B levels? All right, so this slide is going to sum up and answer all of those hypotheses, and because I can do it in one slide, you probably know what's coming. There's no intracellular stores of B at all in the mice or in the humans, and that's consistent with data from the Human Protein Atlas. There's also no change in the factor 13B transcripts, either in the presence or absence of A or when you give A pharmacologically. And we walked every exon because we thought maybe there was a splicing event that contributed to this, but we didn't really see any changes. And we also, in collaboration with Robbie Campbell, looked at ribosomal occupancy. So available messages in the absence and presence of A or in pharmacologically administered A don't change. So there's not an increased translation of existing message going on. And so we eliminate all of these hypotheses. And that left one hypothesis that seemed crazy to us, and that is maybe A stabilizes B, right? So it's not just the B stabilizing A, but maybe this is reciprocal. We had that finding, and then the pandemic hit, and we were limited in getting on the bench. But at that point, we had all of the human data, and we had all of the mouse data, and we had Zoom. And so we began talking with a pharmacologist about how we could go in and model some of these data. And this is the sort of concept we've got here. The idea is that we give A and B pops out, right? And so that could be either because it enhances production of B, or it inhibits clearance of B, and this can be tested sort of mathematically or computationally based on the data. We experimentally had already eliminated the first one. It doesn't seem to enhance production, and the pharmacologic modeling confirmed that. From the pharmacologic modeling, the data were consistent with the idea that, in fact, A is inhibiting the clearance of B, this unlikely third alternate hypothesis that we got to reluctantly by the end. But when we go into the literature, there's a piece of data out there that suggests maybe we should have known this all along. These are data from a study in which they infused A into monkeys. And to me, as a non-pharmacologist, it's a very complicated study, and so we sort of knew about it but hadn't thought about it very much yet. These monkeys are not deficient in A, so modeling this out became very complicated. But fundamentally, from the sophisticated modeling that they did, they found that the A subunit has a short half-life, which we knew. The A2B2 heterotetramer has a longer half-life, which we knew, about 3 days. And they speculated at the time that the B subunit actually also had a really short half-life. And I think because that was so unexpected, this paper really got neglected for a while in the literature. But nonetheless, armed with those data, we went in and did just a very logical experiment, although it was a little bit more complicated than it looks here to do. But we infused A2B2 or B2 into mice, and then we sampled out plasma over time and then computed half-lives. And in fact, the A2B2 half-life in the mice was about 12 hours, and the B half-life was about 4 hours, consistent with what we're seeing that was modeled in the monkeys and probably is going on in the humans. And so that actually suggested, in fact, that not only is B stabilizing A, but the A is stabilizing B. That seems crazy, but what we did, I think, isn't actually the coolest part of the story. What we did is very logical science here. The cool part of the story is here, and this is the stuff we don't know the answer to yet. First, although I hesitate to ever say anything is first, because somebody always came and discovered something or did something before you, and you just don't know about it yet. But to the best that we have been able to figure out in the literature, this may be the first example of an inter-tissue reciprocal stabilization of subunits in a plasma protein. And it sounds a little bit like a baseball stat, but nonetheless, there it is. What that means is that this has got to be some kind of very interesting co-evolutionary phenomenon. What we think we understand from the available literature is that the A and B subunits co-evolved about 500 million years ago with the development of the fish and the evolution of the circulatory system. And again, an expression took place in separate cells in separate tissues. And so what we think is that the interaction, that 10 to the minus 10th binding constant, and the synthesis rates of these two proteins individually with very short half-lives must be remarkably synchronized to ensure that A2B2 is present so that we all have the hemostatic capacity of resident factor 13 in the plasma. And so we're currently talking to a few and seeking additional collaborators in evolutionary biology to help us understand how one even goes about modeling this. But I'm fascinated by this because I think it's just so unexpected and so unusual. All right, so from that, I have no idea what time it is, but I'm at my last chapter now. Is factor 13 a therapeutic target? And if so, what would that look like? So the idea is we have blood, the blood clots, it forms a clot. If we have a dense network, it forms a larger clot. If we have reduced factor 13 activity, it forms a smaller clot. And the clinical translational question to be asked is, can we use this knowledge to convert the large clot into the smaller clot? This is a coagulation cascade that we put together a number of years ago when we were trying to talk about the available hemostatic products, antithrombotic products on the market. The gray are the traditional ones, the pink were the newer ones at the time, the DOACs, and the green were the ones in preclinical trials, although some of those, since we did this, have now progressed through clinical trials and are now advancing quite well. But nonetheless, all of these have something in common, and it's something I said at the very beginning, which is they all inhibit or interfere with the process at or above the point at which thrombin is present and active. So all of these inhibit thrombin and inhibit, therefore, the conversion of fibrinogen to fibrin. And we think that may be the underlying basis for why they are all associated with at least some degree of increased bleeding risk. So if we think about going after factor 13, we see first that it would not interfere with thrombin generation or fibrinogen to fibrin conversion. It would interfere specifically with factor 13 function or behavior in some way, fashion, or form. So we'd get altered fibrin stability, but in that way would be fundamentally different than everything else on the market. And so at least from an intellectual perspective, that's interesting to consider and pursue. If we did that, what would it look like? These are data from the venous thrombosis models that show that as we dose-dependently reduce factor 13, we dose-dependently reduce thrombus size, or this is what is motivating all of the studies that we did. If we take these same mice, factor 13 sufficient hets and knockouts, and subject them to a bleeding model, this is the tail transection model that you heard about earlier, what we see is that the full knockout mice have a bleeding problem, which is actually good because it means the mice simulate what's going on in the humans, because factor 13 deficient humans have a bleeding problem. But what we see is that the heterozygous mice with 50% factor 13 don't seem to have bleeding. And in fact, humans with 50% factor 13 also are pretty good, don't really have remarkable bleeding. So that seems logical. And then when I started giving this talk a few years ago, I got a lot of very good questions about whether we'd increase pulmonary embolism risk because the factor 13 needs to stabilize the clots. So Saravia Katula, again, former trainee, had to develop a new model of factor 13, of pulmonary embolism to look at factor 13 in this situation. What she did is to induce clots in mice, remove the ligature, and then see if an existing thrombus embolizes to the lungs, trying to recapitulate better what goes on in humans than the other models that we have for PE in mice. And when she did that, what we see is that the factor 13 knockouts do in fact get more emboli going into the lungs. But the heterozygous mice do not, suggesting that about 50% of factor 13, and maybe we think even less, is sufficient to stabilize the clots. So we get reduced clot size, no increase in bleeding, and no increase in PE. All right, so that led us to the idea that we need to test this pharmacologically, not just genetically, and we've been on the search for factor 13 inhibitors ever since with mixed success. The problem is that there are existing factor 13 inhibitors, but they all have a short half-life, like 5 minutes short half-life, something that won't work at all for trials, and they have very poor substrate specificity. They inhibit all the other members of the transglutaminase family, which also will not work for any of these studies. So we have a number of these different directions we're going in. I'm going to present one of them here, maybe not even any longer our most interesting one, but I like this one for the shock and awe value of scientists. This is a leech. It's a giant Amazon leech. It's large. You can see that here. It has a very large feeding apparatus that is necessary because it's a hemivore. As a result of all of that, it feeds on very large crocodiles and snakes, and it produces an enzyme that dissolves fibrin to enable that feeding to take place. Because having uncross-linked fibrin is much easier to dissolve, it developed tritogen, which is Welch for 13, and it inhibits factor 13. And it helps prevent those clots from being cross-linked so it can more readily achieve its blood meal. This molecule is actually like hirudin. It's a 66 amino acid protein, a small protein. That one gave rise to a clinical drug, so this one seemed like an interesting target. It inhibits both plasma and platelet factor 13. Remember, it only has to inhibit the platelet factor 13, but it can do both. It has a really good IC50 from a clinical standpoint, medchem standpoint. It has lower potency against tissue transglutaminase, so it's fairly specific. That also bodes pretty well. It doesn't inhibit fibrin formation, so everything starts to look really good, until you get to this part, which, unlike hirudin, it has 6 cysteines that enable 3 disulfide bonds, and if you look across this thing, that's 15 possible isomers. From a medchem standpoint, that's a non-starter. So we've been collaborating with Diana Imhoff at the University of Bonn, who's a fantastic med chemist, and she's been going at this, trying to simplify this molecule so that it can be synthesized and tested. So I'll show just one slide to say where we stand with this now. She's identified at least one of the crosslinks, that's one of the disulfide bonds that's created in this molecule. And she's changed those cysteines to serines. So that bond no longer forms. And by eliminating one of those bonds, suddenly we only have 3 potential isomers instead of 15. And this is something she can work with. So she synthesized and separated these isomers, but for all the trouble of separating the isomers, she didn't actually need to do that because all of them do inhibit factor 13A activity, and we got pretty excited about this. So she sent us these molecules, and we put them in our clot contraction assay, and all of them reduced clot weight really nicely and looked just like T101, so this seemed highly promising. And they have an IC50 that's also not great, but something we can, again, work with. We have done this in humans. We've also done this in mice. And the mice also, again, not a great IC50, but something we can work with. And we see the potential that we can translate from human systems to mouse systems, which is what you want. The next hurdle that we have run into that we're still working to solve is that its half-life is terrible. So we're still working on this. And I always think about this, I put this at the end of a talk. It's really a downer way to end a talk, but it's the real way to end a talk, right? And so every time we meet a challenge, we have to come up with another question or another experiment, and that's where we are. All right, so to put all this in context, I showed this and walked you through at the beginning of it. For everything that you've heard for the last 30 minutes or so, we've added this tiny piece here, which is that factor 13A activity participates in the consolidation of these clots into these small clots that occlude blood flow. And so from this, we speculate that if we can target this, if we can inhibit factor 13, the way it interacts with fibrinogen or fibrin or some other facet of this, we may be able to release the red blood cells from that clot, making smaller thrombi that may be less likely to occlude blood flow. All right, so the work that I presented is work that spanned a number of years, and so there's a lot of people that have contributed to these studies. I've tried to highlight in bold current and former trainees in the lab that have really contributed in some way to this story. There's a lot of names now, so I won't read them all out to you, but I'm incredibly grateful for their persistence through what you hopefully understand is a lot of negative data as we disproved a lot of hypotheses. But they've been fantastic, and I appreciate you all as well. Thank you for your attention, and I'll be happy to take questions.
Jose Lopez from Seattle. Nice talk, Alisa. I have a couple of questions. One thing that I didn't understand, maybe you explained it and I missed it, but you were talking about B stabilizing A and A stabilizing B, but I didn't understand why you have, when you have AB, you have twice as much B as A. I think that's what you said, that the amount of free B is the same as the amount of A2, B2.
Yeah, I'm not going to be able to explain this using any kind of sophisticated terminology, but the idea is that B is synthesized at a really high rate, so we have a given amount that's present constitutively. It's getting rapidly synthesized and rapidly cleared. But when you have A and you're able to stabilize at least part of the B that's there, there's a different clearance mechanism for that particular component. So then you have the free B that's already there, and now you have the complexed B as well, and it's the combination of both of those that effectively doubles it. And again, I can't run the numbers for the math. That's why we called in a pharmacologist to help us do the modeling of this. But the doubling occurs from the fact that it's now in sort of 2 plasma compartments, one in a bound form and one in a free form. Keeping in mind that I want to watch my own terminology, free doesn't mean that it's there by itself. It means that it's bound to fibrinogen. Both of these are bound to fibrinogen. But we know that getting rid of fibrinogen doesn't change any of this. So that's not part of the equation. It's just true.
Is it possible that the A is binding to a clearance receptor that clears B?
Yeah, we don't know what clears A2, B2. We don't know what clears B. I mean, we don't know any of the clearance mechanisms. It's something that is part of maybe my next chapter. Yeah. And so, yeah, it could be either one. One of the things we were wondering about is if either of them have an exposed epitope that in fact does recognize some type of receptor, maybe the same or maybe different ones, but when they form a complex, they sterically block the availability of each of their clearance receptors so that what's left is a completely different clearance mechanism that has to be mediated by something on the exterior of wherever that interaction is taking place. It's all speculative right now.
Thanks. Second question. So the mice that lack factor 13, you said that they had more pulmonary emboli. you would suspect that those emboli would be smaller and easier to lyse and may not be such a big problem. Did you see any kind of like clinical signs in those mice that they were getting more emboli?
Yeah, we tried to look at that. It turns out it's really hard in mice. It's hard to say when they're hard bleeding. And we looked at ventricular function. The best data from that, I'm really glad you said it. The best data for that is increased PE is not one of the clinical presentations of a factor 13 deficient human. It's weird. We pointed to that as we were beginning the study because the problem is that there's just not that many factor 13 deficient individuals. I think it's estimated like 1,200 worldwide, many of whom are treated. So the idea of trying to find enough untreated factor 13 deficient individuals to be able to ask the question in humans, does factor 13 deficiency increase PE, or even if it does increase PE, are they asymptomatic and not problematic because as soon as they hit the lungs, they just dissolve? I kind of think it's that. I think from the mice, it's probably logical, and we're not the only one. Peter Gross has done some studies on this too, to suggest the emboli are getting to the lungs. I just think they're not a problem because they're so rapid. They're small, they're not occlusive, and they're so rapidly dissolved. And that's why we don't have any data from the humans. In fact, the absence of data is probably speaking louder than what we're seeing in the mice. Yeah.
Hi, Danielle from University of Toronto and Heyu Ni's research group.
Oh, yeah, hi.
Really great talk. I was wondering if you know the role of platelet factor 13 if it's not in the canonical fibrin strength mechanism.
We don't. We've been looking at this, and the way we've gone about doing it is rather than stumble around and try model after model after model to try to figure out how to do this, and knocking out factor 13 in the platelets is very hard because it also eliminates the plasma at the same time. It's a very complicated series of crosses, and it doesn't really get you anywhere. So what we've done is to even try to understand how does the factor 13 even get out of the platelet in the first place? Because if we knew the conditions under which the factor 13 stays in or comes out of the platelet, it might narrow the field for the clinical situations in which it's relevant. Unpublished data, we're still writing it up right now. It looks like 80% of the factor 13 that's present in the platelet never gets out. Maybe is never accessible to anything that's trapped within there. So about 20% comes out probably with or part of extracellular vesicles and seems subjected to all the normal factor 13 biology that happens after that. The stuff that's in the platelets is like incredibly stable. And so one of the thoughts is that maybe the intracellular component in the platelets is, I don't know, vestigial or regulated in the platelets or similar to the factor 13 that's present in leukocytes, and it's more involved in intracellular processes than extracellular processes. But we don't have any data to tell you what that is per se. There's a few studies out there, but we don't really know.
Thank you.
Sure, thanks.
Can I ask a quick question about your model?
Yeah.
Sorry, your PE model. I was just curious what the time course for the embolization occurred in the mice.
What we did, that was such a messy model. It's really hard. So we opened up the mice, ligated the inferior vena cava, closed up the mice. 24 hours later, reopened up the mice, took off the ligature, closed the mice back up, and then came back 24 hours after that, harvested the lungs, and looked for evidence of emboli in the lungs. It's a really hard model from a surgical perspective. Brian Cooley helped us with all of those experiments. I don't know anybody else that really could have done it at the time and done it as elegantly as he did. We still had, trouble getting all the mice through everything, but that's the timing. We weren't able to really look. We tried labeling things and looking in sort of longitudinal type imaging, and it just didn't work well. So we ended up sectioning through the entire lung and counting clots. It was challenging, but that's what we came up with.
Yeah. Raymond Adley from Bloodworks. I wonder if you look at VWF, if VWF has any role of retention of RBCs on those deep vein thrombosis models.
We looked at that. There's a bunch of data that's pretty cool and intriguing to suggest that VWF can get intertwined in the fibrin strands and probably increase the strength of those fibrin strands. We looked at that in von Willebrand factor deficient mice. We even did it by replacing, it was complicated, to replace the 8 because that's also down in the von Willebrand factor mice. And we did it by tag and looking at clot contraction and to see if there were any changes. And it doesn't. In our hands, we can't find any evidence of von Willebrand factor contributes to the red cell retention. And that was surprising to us. So we published that, I think, in ATVB, and we think no.
Try other models and other than ligation model, like electrolytic injury model or stenosis model?
We've done, yeah, we did, when we did the original finding, we did stasis and we did stenosis. We have done a ferric chloride injury model. Factor 13 doesn't seem to contribute to the way the clots are forming, at least the occlusion times in that. Although if you inhibit factor 13 completely, the clots are easier to dissolve, and Merck did a series of trials in rabbits and dogs in different models way back in the 90s that our data are consistent with that. We've done some electrolytic modeling, but we've stayed away from that mostly because those clots are not the red cell-rich clots that recapitulate the histology of a VTE in humans. So we tend to gravitate towards the stasis and stenosis models. And of the two, unless we see a reason to go after stenosis, like in the platelet study that Wolfgang and I did, we tend to go for stasis because it's a higher hit rate and you can kill less mice. Yeah.
Thanks.
I have a question from the virtual audience. It's from Madeline. She says, awesome talk. I was just wondering if you have tried siRNA against factor 13 as a way to knock it down.
Christian Kastrup did that in a really nice study published in Blood a couple years ago. The idea is, yeah, it dovetails exactly with what we have. That if, at the time, if B is regulating A, so this preceded our current study, then if you could siRNA down the B, you could reduce the A, and so you may be able to do that. And he actually showed that really well with all of the biochemistry that he does, and some models. I think he actually, the one he didn't do was venous thrombosis, but he did it with the rest of them, and so yes. And yeah, great question, and it can be done.